Today we'll study network pharmacology. In today's class, what are the things we will study? First of all, introduction of the network study and definition of the network pharmacology, and what are the fundamentals, theoretical fundamentals, data sources, processes, as well as the big data analysis, how this network pharmacology, the big data we are analyzing. And next one, network construction and interpretation, predictions, and then the network analysis, how we are analyzing, and then network visualization, and lastly application of the network, as well as in Ayurveda by taking one simple example how this network pharmacology is helpful, that we will study in today's class. Okay, first of all we will study the introduction. What is network pharmacology? Okay, it is a evolving science. In today's life it is one of the evolving science. Here it is a powerful tools. Nowadays network pharmacology is using as a tools in drug discovery and development. Okay, whatever the new drug discoveries and development are going on, this is the science that we are using. This is network pharmacology we are using as one of the tool. Here previously the traditional method focus on single drug and single targets, means the single drug that we are using and single target, that is traditionally that we are using in the drug discovery and development. Here the network pharmacology examines how the multiple drug affects multiple target in the network. Okay, in the whole network. Okay, here this multiple drugs or a single drug, that is how multiple target that we will study in this network pharmacology. Okay, first time this is told, or this science is given by Hopkins in 2007. Okay, now the definition of the network pharmacology. Here the network pharmacology is a study how drug interact with multiple biological targets within complex system rather than focusing on single drug. Okay, what is it? Definition. A definition tells it is a study how drug interact with multiple biological targets. Okay, not here single target. This is a study. Network pharmacology is a study how drug interact with multiple biological target, okay, within our complex systems rather than focusing on single target. Okay, traditionally what we are using, the single drug and single target we are using. This network pharmacology is a study. It is a study how drug is interact with multiple biological targets within the complex system rather than focusing on a single target. Okay, now this science is a combination of it is a combined system. Here what are the sciences involved? The biology, bioinformatics, pharmacology to improve the drug discovery and treatment. Okay, it is a network pharmacology is a combination of the systems, biology, bioinformatics and pharmacology. All these sciences are involved in this network pharmacology. Okay, why these combined disciplines we are using for the drug discovery as well as treatment purpose. All these sciences we are combining. Okay, this is a new branch. This is called network pharmacology. Okay, now the theoretical fundamentals. Here this network pharmacology explore the drug action on biological network. Okay, here this network pharmacology explore drug action. Okay, how the drug is action on biological networks, the whole network. Here they emphasize the multiple target interactions. Okay, not only the single target, multiple, how it is targeting, and the system level understanding the disease. Okay, by systematically we are understanding the disease. Okay, this is the fundamentals we have to understand before we have to study this network pharmacology. It is the network pharmacology is a explore drug actions on biological systems or biological network, emphasize multiple target interactions and system level understanding of the disease. Okay. Now the next point is data sources. Okay. Here network pharmacology required some data sources. Okay. It is already, we studied, this is the bioinformatics science and biology. It requires some the datas. Okay, datas we are using to study the network pharmacology. Okay, what are the sources available to study this one? The PubMed and Impart, PubChem and Binding DB and DisGenet and KEJG strings as potential source of information for drug targets. Okay, all these sources we are using as information for drug target and disease, gene and protein interaction and single pathway. For study all these things we require all these databases. Okay, like apps or webs, these we are using. Okay, these apps or these data sources we are using to study the network pharmacology, in the network how the drug is acting. Okay, that we'll study by using these sources. Okay, now we'll study the process. Okay, how, for example, if you are using any drug discovery or development, okay, how we have to utilize this network pharmacology. The process one by one we will study. It requires total 5 steps for this process. The first step in the process, the first step, we are doing the screening and identification of active constant within the drugs. Okay, by using one data source. Okay, that is by using PubMed or PubChem or Impart. These data sources we are using to screen and identify the active constants. Okay, suppose one drug it is there, which active constant it is present. For that purpose, just we have to do simple, just we have to open this PubMed or PubChem. After opening this database, or you can call it a search engine, just you enter the drug name. Okay, if you enter the drug name, then what are the constants, chemical constants or active constant it is present, it will show. This source we are using in the first steps. Okay, first step here we are using. Suppose the Bakuchi it is there, Soralia corlifolia is one of the drug. Okay, suppose if you search the Soralia corlifolia or Bakuchi in this search engine, then it will show what is the active constant, Soralin or other whatever the active constants. Okay, that will show. Okay, like this the Terminalia arjuna it is there. Okay, that suppose if you search this, if you put in this search engine like PubMed in PubChem, then it will show what are the Terminalia arjuna, what are the chemical constants are present or active constant it is present. Okay, like terminalin, arginine, all that it will show. Okay, this is the helpful. In the first step, what we are using, that we are screening and identifying the active constant within the drug. Okay, the next step is target identification. Okay, now in the first step we search what is the drug, the particular drug, what is the active constant it is present, that we studied. In the second step, okay, what is our aim? Okay, where we have to target, which area we have to target, or which protein or which gene we have to target, that tool we are using. Okay, like data binding. Okay, suppose one active constant it is there, where it is the combining, okay, where it is binding, that will, in the second step we will study the target identification. This tool or this database is helpful for the finding of this database, this identification of the target. Okay, very good example of this search engine is Binding DB. Okay, if we put the particular drug or constant, then which target, suppose if you put the one drug name, or active constant, okay, it will show, it will where it will combine. Okay, that combine, take for example, suppose the one disease if you are taking, that is Arjuna, if you are again taking the same drug Arjuna, then it will bind with the, that is cardiac muscles or cardiac diseases, that will show. This binding DP is helpful for the, if you put the drug or any the active constants, this app or this database is helpful for the showing the where it is combining. Okay, this active constant is combining with the which target, that will it will show. Okay, this is the second step we are using. Okay, come to the third step. Here third step, the disease gene identification. Okay, if you are finding the targeting particular disease, then which gene is involved or which protein is involved, that is we are using, by using this app or this database, this database, by using this datagenet or this search engine, by using this one, we can easily identify the which disease gene is involved. Okay, for the producing the particular disease, which gene is involved. This source we are using in the third step. Okay, now come to the fourth step, that is gene ontology. Okay, this is the gene ontology. This is a philosophically we are thinking. Okay, philosophically we are thinking that which gene is, how it is acting. Okay, here fourth step, the gene ontology. Here gene ontology means it is will give the philosophical answer how this gene is involved. Okay, how its pathway it is there, how it is revolves to target the diseases. Okay, by different targets, how it is using. Here in this search engine, what is the tool we are using? KEGG pathway. This tool we are using for the searching the philosophical answer of the how the gene is acting. Okay, how the gene is useful. Now the fifth step is network construction. Here we are using, by taking all these sources, we are using this map, or we are constructing a network. Okay, whole network from the starting point to the end point, what are the things are involved. Okay, here this network construction, the source or the database we are using, Cytoscape. This database is helpful for the network visualization as well as analyzing software. This software is we are using for the network visualization as well as analyzing this whole network. Next one is the string. This one is the construction of protein interactions. Okay, how this one protein is interact with other to produce disease, or as well as for the treating of the disease. Here this string is helpful for the constructing the protein interactions. Okay, this is the software or search engine we are using. Okay, these are all the 5 steps we are using in the process of the network pharmacology. Okay, the next one is the big data analytic. In this step, whatever the big data it is there, handling the large data sets generated by high thoughtful technology. By using the high thoughtful technology, we are plan to try to understand, okay, what is the meaning of that data set generated high thoughtful technology, by analyzing what is the meaning, okay, how this particular drug is acting by using the different networks. The analyzing purpose or understanding purpose, this we are using. Okay, the next one is the network construction and interpretation predictions. Okay, here the constructed network can be interpreted. Whatever the network now it is in the cytoscope or string we are constructed here, that is helpful interpreted, or that is the interpreted understanding that network. This is helpful for the predicting potential drug target. Okay, whatever the network it is there, this is helpful for the predicting the how this drug is targeting the particular thing. Okay, that is helpful. Next, by using this network, that is helpful for the mechanism of action, how the drug is acting. To understand the mechanism of action of mode of actions, this is helpful. The next one, potential side effect. Okay, in the early stage only we can identify the is there any side effect it will produce. That also helpful by the predicting this network pharmacology we are using. Okay, the next step is network analysis. Okay, here it is a, in the previous the data analysis, now that it is network analysis. The network analysis, the extract meaning inside, whatever was the hidden meaning, that is this is helpful for the extracting the meaningful insight whatever it is there, and the shortest pathway analysis purpose. Okay, what is the shortest pathway? Okay, by understanding the whole the network, this helpful for the shortest pathway is there any, that also we are analyzing here. Next one is the centrality measures. Is there any centrality measures are there? That also we will study here. And next one is the network modifiers. This is also helpful by using the network analysis. Okay, this is about the network analysis. The next step is network visualizations. Okay, the network it is there for visualizations. It is a tool like, okay, the tools like that cytoscape for the effective communicating complex network data. Okay, just now we discussed that is the network visualizations. Okay, by using this this cytoscape, this is a tool like a tool effectively communicating the complex network, whatever the drug it is in the complex system, how it is effective, how it is doing actions, all this understanding purpose, this is cytoscope is helpful for the visualizations. Okay, the next one, applications. Application uses. What is the use of this network pharmacology or application? The first thing, why we have to study the network pharmacology, because first is identify the new drug targets. Okay, the identify the novel drug targets. Okay, here this is a very good science for the identifying the new drug targets. If new drug, if you are doing research, the what is the target it is there, that is helpful. Next one is that is repurposing the existing drug for new interactions or new indications. Okay, whatever the oldest previously the drugs we are using, okay, the particular treatment in that we are using, is there any other uses are there? Okay, that drug is helpful for the treating any other diseases. Okay, that also this network pharmacology is helpful. Okay, that is repurposing existing drug, whatever the existing drug is there for new indication. Is there any new indications? Okay, the drug it is there. Is there new benefits? Is there doing any treating the new disease? That also understanding by using this network pharmacology. The third one is understanding drug mechanism of actions. Okay, this third benefit or third utilization is that is this network pharmacology is helpful for the understanding the drugs mechanism of actions. The next one, fourth one, is the predicting potential drug side effects. In this fourth step or the fourth benefit is there any side effect it is there? That is also we can predict in the early stage or in the first stage by using network pharmacology. The last one is the fifth, developing personalized medicines. Okay, different people's the different body structures. Okay, if you're taking the Ayurvedic Vata Pitta Kapha, the different prakritis are there. Okay, by taking all this, considering all this, we can use the developing a personalized medicine strategies. The personalized medicines we can prepare by using this network pharmacology. Okay, the next one, last one, network pharmacology and Ayurveda. Okay, how it is helpful for the Ayurveda network pharmacology. Here, network pharmacology helps to unravel triphala effects by mapping how it is multiple compounds interact with various targets within the body. Okay, by using this network pharmacology, it will helpful for the open the triphala effects. Okay, it will explore the triphala effects by mapping how it is multiple compounds interacts with various targets. Okay, here it is one example we are taken, that triphala. Okay, in this three phala, here the multiple compounds we are using. Okay, these multiple compounds, how these are affecting or how they are acting on the different targets. Okay, within these targets, various targets within the body. That is, we will understanding by the using of the network pharmacology. Okay, this is all about the brief information about the network pharmacology. Okay, hope you will understood. Okay, thank you for watching. Please subscribe channel.